Youth Biolab Jeunesse.
Hi, I'm Steve.
I'm Megan.
And I'm Angelica.
We're from the Youth Biolab at the Albrechtsen Research Center at St. Boniface Hospital. We all know the job of the hospital is to treat patients. Here at the Research Center, our job is to study disease. If we ever want to cure a disease, we have to understand it first.
When you think of scientists studying disease, you probably think first of things like cancer or more recently, COVID-19. We don't study either of those here. But there are research scientists over at the National Microbiology Lab who are studying infectious diseases like the flu or COVID-19 or Ebola, where they have special equipment that can protect them from the germs that they're working with. There are also groups of cancer scientists working over at Cancer Care Manitoba in Health Sciences Centre, and it's their job to study cancer. We like to keep scientists together so that they can work as a team to come up with cures or treatments for these diseases that we see.
At the Albrechtsen Research Center, we have three big interests. On our fourth floor, we have the Division of Neurodegenerative Disorders. This is where we study diseases that affect the brain and nervous system, just like Alzheimer's disease and type 2 diabetes nerve damage. On our third floor, we have the Institute of Cardiovascular Sciences. This is where we study diseases that affect the heart and cardiovascular system. On our second floor, we have the Canadian Centre for Agri-Food Research in Health and Medicine, and this is where we study food. They research foods that are really good for us, some that might be bad for us, and some that might be so good that they act like medicine.
Our lab, the Youth Biolab, was created so that we can share some of the things that we do here in the Research Center with you. And hopefully we can help you learn a little bit more about how medical science works and understand a little bit more of the science of your own health. I'm here at the entrance to the Research Center. It's the first week of September 2020, as everyone is preparing to get back to school. As we all know, 2020 has been a very different year, where we're all focusing on keeping ourselves and our friends and families healthy in the face of the COVID-19 pandemic. Just like at your schools, the hospital and the Research Center have been taking all kinds of measures to make sure that we can do the work we need to do and be safe while we do it. We've been masked up here since June, we've been distancing, we've been sanitizing our hands, and we know it's probably looking the same around your school. Signs, arrows, and all kinds of new rules. This all takes a lot of getting used to, whether you're at school or at work. There's a lot to understand when it comes to dealing with this virus, and sometimes it seems hard to keep up. Information changes as scientists learn more about this coronavirus all the time. The rules seem to keep changing, but that's a really good thing. This means that scientists are learning and understanding more about this virus every day, and they are letting us know how we can keep each other safe. Now that I'm back down in the lab, where I'm not in close contact with other people, I'm allowed to take my mask off because I know I'm not going to be within 2 meters of any of my coworkers. If we are working together, I'd put the mask back on. So I'm going to take it off to continue with the video. First thing you always want to do when you're taking off your mask is make sure you get your hands clean. So either soap and water, a good long wash, or some hand sanitizer. So I'm going to clean my hands with the hand sanitizer and really rub it between my fingers, making sure I get my fingernails, my palms, and every surface of my thumbs between my fingers and my fingernails. Now I know that my hands are clean and I'm not going to be putting any germs near my face as I go and take my mask off. The big rule of masks is don't touch it. We really, if we want to take it off, just loop our fingers behind and peel it off without touching our face. This one's been used. It's a single-use mask, so now I've got to get rid of it before we continue. Since this virus popped up in late 2019 and began to spread around the world in 2020, scientists have been working to understand everything about this new coronavirus. Since the start of the pandemic, thousands and thousands of studies have already been done as we start to understand how the virus infects people, how it spreads, and how to protect ourselves from it. These are not easy problems to solve. You can imagine any problem like this like a giant puzzle. When you sit down to start this puzzle, you have no idea what it's really going to look like when you're done. So the best way to approach it is to focus on the small parts. We share everything we learn with the world by publishing our results so other scientists can see what other puzzle pieces look like and start piecing it together. As we do this, we take shape and we can understand the bigger picture and start to understand the whole puzzle. This isn't super easy. Sometimes you think you have the right piece and it turns out not to fit quite right. Surely though, the efforts of labs around the world will slowly help find the solution. When we want to understand how to fight the COVID-19 pandemic, we can look at some of these puzzle pieces. We know that washing our hands is a great way to prevent all kinds of germs from infecting us. With this coronavirus, it seems like distancing ourselves from each other by at least 1 meter is effective, and even more effective if we can stay 2 meters apart. If we can't do this in school or in smaller spaces, well, what do? This is where scientists think that mask wearing will help. So here's a couple of studies from the last few months that are looking at mask wearing. This group of scientists used complicated math to show that mask wearing in communities can reduce infections and deaths. Other groups of scientists studying mask wearing have shown that masks might help protect both the mask wearer and those around them. All these groups tell us that much more research is needed, but remember, we're just at the start of this puzzle. A year from now, we'll know much more. That's the way the science works. Based on how the puzzle has come together in the last 9 months, it seems that hand washing, distancing, and mask wearing are the best tools we have until we have a vaccine. Here at our research center, we don't have labs that study infectious diseases like this coronavirus, but we have a bunch of curious scientists who wanted to get involved when this pandemic started. Some of our scientists here have started looking at how their research skills could help, and they turned their attention to this question of masks. Think about everything you've heard and seen about masks, and you'll realize there's a lot of questions to be answered. What material is best? How do you wear it properly? Can we reuse our masks? When should I be wearing it? There are so many different types of masks out there. You might have cloth reusable masks like the ones you're probably wearing at school. You might have 3-ply surgical masks like the ones we wear here at the research center. There's also N95 masks, which give you an extra level of protection. And then there's all kinds of other masks. And it's hard to know which one you're supposed to be wearing and when you're supposed to be wearing it. Here in our lab, we were curious about this question too. And we thought we'd try a quick demonstration to see how well the masks that we wear to work every day filter out germs. We're wearing these 3-ply medical masks, sometimes called surgical masks. 3-ply means that it has 3 layers that are designed to trap any droplets that come out of your mouth or nose that might contain any germs. If you look closely at this one that I've cut open, you can see there's a middle layer of polypropylene that can be used to filter out those droplets from our nose and mouth and prevent germs from escaping into the lab. So what we're going to do is we're going to use these little dishes that are filled with something called agar. Agar is a food source for bacteria. So if bacteria come out of my mouth and land on this stuff, they'll grow overnight, and we should be able to see tiny piles of little germs called colonies. This yellowish one is called LB agar, and this brown one is called chocolate agar. It's not chocolate, it's just called that because of its color. Bacteria like to eat different things, so if we give them a couple different types of food sources, we should be able to catch some that live in my mouth and my nose. Some of those bacteria could maybe make you sick, but most are probably harmless. We all have bacteria in our mouths and noses. One thing to note is that these plates will not catch viruses. They might grow bacteria or fungus, but a virus like the coronavirus can't grow on this stuff. It actually needs living cells to grow. So we need a whole totally different testing system to detect any coronavirus that might be in my mouth or my nose or my lungs. So what we're going to do here is I'm going to talk at some of these plates at a distance of about 10 centimeters. I'll do it without a mask first, and then I'll try it with a mask and see if there's a difference in the amount of germs that end up on these plates, the amount of bacteria. Then what I'll do is I'll cough on these plates from a distance of 10 centimeters. Again, without a mask and then with a mask. And we should try and see a difference as those bacteria start to grow on those plates. Now I'm going to mask up and we'll try it again. So hopefully, or hopefully not, some germs have come out of my mouth in those little droplets and have landed on those plates. One thing that'll help those germs grow is to keep them nice and warm overnight. Bacteria love growing at a warm temperature. That's why they like to live in our bodies too. So we're going to take these guys and we'll put them in an incubator that'll keep them at body temperature overnight, and then we can check them in the morning and see if anything grew. So we've waited overnight and I got our plates back out of the incubator to see if any of the germs have grown onto these dishes. Remember, we did some with no masks and talking, and some with masks and talking. We did coughing with no mask and coughing with a mask. So let's have a look at the results. Overnight, any germs that came out of my mouth or my nose and hit this plate would have started to grow and form those little piles called colonies. So first, let's look at the plates that I made of me talking with no mask. If we look closely at the agar, we can actually see some very small colonies. They're a little hard to see on the LB plates because they're more of a clear agar. But if we look at the chocolate agar plate of me talking with no mask, there's some very obvious colonies on this plate that have come out of my mouth or my nose and landed on the agar and grown overnight. You can see they almost have a little bit of color. Some of them almost have a greenish color. When we turn to the masked plates where I was talking, I can't see a single colony. Sometimes it helps to turn it up to the light, but I don't see any colonies on this one. And if we look at our chocolate agar, I can't see any colonies on this. If we turn to our coughing plates now, if you think about it, there's obviously more droplets, more things coming out of my mouth if I'm forcefully coughing than if I'm just speaking. So what would we expect? We'd expect more colonies growing on these plates. If I look at our agar plate, I can definitely see some colony growth here. And let's turn to our chocolate plate, tons of colonies. We can see some really large ones. If I were to count these up, I'd say there's about 50 colonies on this chocolate agar plate. So that's coughing with no mask. Definite growth, definite germs coming out of my mouth. That's what's going on every time you're coughing and not covering. Let's look at what a mask does for us here. If we look at our LB plate, I can maybe see one tiny colony, and that might even be a piece of dust. Almost nothing made it through that mask onto my LB plate. And if we check our chocolate agar plate where things should be a little more obvious, I don't see a single colony there. So this is a pretty simple demonstration of how effective a mask can be, of protecting other people from the germs coming out of my mouth. That mask is effective enough at that close distance to block most of those germs. Now remember, viruses won't grow on these plates, so maybe viral particles, which are much smaller, could get through, but that mask is doing something. We can see that here. Now this was a fairly simple demonstration. To make this into a real experiment, we would have to repeat it a bunch of times and be a little cleaner about it to make sure that no other bacteria or fungi from the environment were getting on those dishes. We could also test different distances and other types of agar to see what's actually coming out of my mouth and nose. There's a ton of work that goes into answering these kinds of questions accurately. So maybe we should find a scientist who has done some bigger studies on mask use. Here at the hospital, mask use is really important to protect patients and staff, and we go through a ton of masks every day. These ones are called N95 respirator masks and are one of the best single-use masks for filtering out virus particles, even better than the surgical masks we just tested. We go through about 400 of these masks every day at the hospital. So an important question came up. Is there a way to clean these masks we use so that they can still be safe to reuse? We're going to head upstairs to meet Mike Shubert, who did a study like this, and he can tell us everything that they did. So we're here on the 4th floor at Dr. Mike Shubert's lab. Mike, together with a team of researchers from the research center and from the hospital, worked on a study on masks this summer. It was published in July 2020. And what they did was put out a paper called N95 Mask Reuse in a Major Urban Hospital, COVID-19 Response Process and Procedure. When you publish something like this goes out so other scientists around the world can learn what you've learned and start piecing that puzzle together. Let's go into Mike's lab and see if he can tell us a little bit more about what they did in this study. All right, so we're here with Dr. Mike Shubert. Mike's a professor in the Department of Physiology and Pathophysiology at the University of Manitoba. And Mike runs this lab here. You're the
Molecular Pathophysiology Lab.
Molecular Pathophysiology Lab. So what does that mean?
It means we study how disease happens at the subcellular level, proteins, DNA, that sort of thing.
But you're mostly a heart scientist. So what got you into putting together this study here on masks. How'd you end up studying masks?
Well, I was asked to take part in a task force that was looking at ways to respond to the COVID-19 situation way back in March, when we really didn't have an idea what the situation was going to look like in Manitoba. So one of the ideas that came out of that was, is there a way to reuse single-use N95 respirator masks?
Right. And so we had a look at kind of different types of masks down in our lab. We talked about, the masks that we're wearing and cloth masks that kids might be wearing. Why N95s? Like, why are they important?
Yeah, it's very confusing because there's a lot of different kinds of masks out there. The N95s are specifically geared towards folks like those working in a hospital, where you might have a doctor or nurse working with a COVID patient, and they need to ensure that the doctor and the nurse doesn't become sick themselves working with these patients. So the N95 masks are designed to minimize the risk of that happening by making sure that the air that they breathe in is sterile, that it's clean and is not going to transmit the virus to them. The masks we wear every day, such as the ones we have on right now, are a little bit different. They're not built the same way, and they have a little bit of a different focus. So these are more about breathing things out onto others. And it's the idea is that if you're sick out in public and you don't know it, by wearing a mask, you reduce the risk that you'll spread it to somebody else. And if everybody wears the masks, then we all actually come out much safer as a result.
So I know you came together with a team of other people from here in the research center and also some folks over in the hospital. And what was the study you set up? Where'd you come up with the idea? What was the main idea of the study?
Well, so as I mentioned, the masks are generally designed to be single use. That's the intent, that you wear it, you throw it away when you're done, you take another one at your next shift, or if you go into a different area. The problem is that we didn't know if we might run out of these masks. We knew that supplies became difficult to get, not just here in Winnipeg, but globally and across the world. At the time, there had been a little bit of work suggesting that brand new N95 masks could be sterilized up to 10 times, and they still worked. What we found, though, was that the masks that had been sterilized that way had not actually been used. And our question was, well, what happens if you actually wear it when you're in the hospital? You're sweating into the mask, you're breathing through the mask, all these things, it's deforming to your face a little bit. Does that affect your ability to reuse the masks? And the answer was yes. So we can sterilize the mask, but it turns out we could only do it one time. After that, the mask became degraded enough that we couldn't keep the person wearing it safe.
What kind of sterilization technique did you use over at the hospital?
So we used a technique called autoclaving, which is basically raising the temperature of the mask under high pressure for about 30 minutes. So we raise it to 121 degrees Celsius, high pressure, 30 minutes, and then we let them cool down, and then we actually tested them to see if they still worked. And that was the key point, was doing the testing. We do a very particular kind of testing in this called a fit test. Okay. So what a fit test basically does is you have somebody wear the mask, they're in a room that has basically a salt vapor in the air, and you sample air outside the mask and from inside the mask, and you compare those two. Now, if the mask works, you'll measure salt vapor outside the mask and none inside the mask. Wow. If the mask is not working, you'll start to measure salt vapor inside the mask. So you put the mask on, you do this test, and during the test, you do things like nodding your head up and down, turning it side to side, speaking loudly, things that mimic wearing it in the real world. And that's what our study was really about. So we had people wear the masks for a day during their regular work shifts. We took the masks, we sterilized them by autoclaving. Then we tested them out the next day with this fit testing. Then we did it again. And it turns out after that first sterilization round, everything was great. But after the second sterilization round, some of these masks started to fail. So our take home was, unfortunately, we can only reuse them once, but being able to reuse them once is a huge gain over not being able to reuse them at all. And it does allow us to stretch our resources.
You can almost double the number of masks we have available to us. And the other part is, like we said, this paper, this study gets shared around the world. So maybe other researchers and other hospitals halfway around the globe can now kind of see what you've done and what you've shown, and they can use that to help maintain their own supplies. Because this is a global pandemic that affects every single hospital across the globe, right?
That's exactly right. And really our paper, I think, is a way to tell others how to do their own studies, because we used a particular kind of mask. Another hospital might use a different kind of mask. They may use a slightly different autoclave. And all these variables have to be tested to see, in your specific hospital, your specific doctor's office, or whatever it is, does this technique work? So our paper is really a how-to guide for others to do what we did.
Sure, and that's what we were talking about before also was, that giant puzzle that we're working on. Mike has his one little piece of that mask puzzle, and there's other groups around the world studying masks, maybe these masks, maybe different types of masks, maybe different sterilization techniques, and we're starting to see what the best way to use masks is.
That's exactly right. There are a number of ways to sterilize masks. So there are things like ethylene oxide vapor or hydrogen peroxide vapor. There's a number of things that have been tested, and a lot of them work. It turns out it's actually not too difficult to kill the virus, but it is difficult to keep the masks intact after doing that. And that's really the balancing act that we're facing.
So you're normally my go-to heart expert. Now you're my go-to mask expert. We've talked about, there's a whole bunch of different things we need to do to keep ourselves safe, washing our hands, keeping our distance. Just to sum it up for people who are watching, why is mask wearing important? Why is doing studies like this? Why are doing studies like this? Why is this important?
It's important so that we understand what we're doing and why we're doing it, and if there's ways to do it better than we currently are. So at the very beginning of this, we had limited information. We knew from other outbreaks and other viruses and things, some of the general techniques that work well in an outbreak. But every virus is a little bit different. Every outbreak is a little bit different. And so we had to start asking questions, well, what's unique about this? What's different? How do we test that? And how do we assure ourselves that what we're doing is the best possible course of action? And honestly, as things went along, our understanding changed. And so the instructions we started to hear about what to do and what not to do started to change a little bit as well. The fundamentals are always the same. Washing your hands, keeping distance, those things are really, really critical. Early on, it wasn't clear, did we need to wear masks or what kind of mask? How important was that? But I think the data has really come out. Now, there's been a lot of studies in the last few months. There's been just a huge flurry of activity in this area to ask these questions. Do masks make you safer? Yes, there's absolutely data out there that says masks make you safer. And the more people that wear them, the greater the margin of safety for everybody involved. And just get used to the new normal, as it were, in what we have to do to not only keep ourselves safe, but keep those around us safe.
Well, fantastic. Thanks for sharing what you've learned in these studies and keeping us up to date on what we're learning about masks and where we need to go from here. I just have one last question for you. How effective do you think this mask would be?
Well, I think it'll help make sure people stay away from you. So in that way, it may be somewhat effective. Perhaps better if you put one of these over top of that as well.
Very good. Thanks, Mike. Thanks for tuning in to Healthy Curiosity. Stay curious.